Welcome back to so today we are discussing the SQs from the photosynthesis and the respiration chapter. So till now we have finished with the transportation in plants, the mineral nutrition, and the enzymes, and in the photosynthesis, two questions we have finished. That is the structure of draw the leaf table diagram of the chloroplast and the differences between C3 and the C4 cycles and the plants. These two questions we have finished. Now we will go with the next question, that is describe I'm having some other mind in my mind going with that. Hello, audible? So welcome back to Botany second year students. So today we are going to deal with the remaining SQs from the unit 1. That is the 4th chapter and the 5th chapter SQs. After that, we'll go with the 6th chapter SQs. That is the plant growth hormones. Okay. So in the 4th chapter, we have finished the neat labeled diagram for the chloroplast and the differences between C3 and the C4 plants. Now we will go with the next question, that is describe the C4 pathway. So what is the C4 pathway and how to write the answer for this question? So C4 pathway, it's not the pathway actually, it's a C4 cycle. Hatch and slack cycle. C4 cycle is also called as the hatch and slack cycle. This is going to be seen in the tropical and the subtropical plants where they can tolerate the temperature up to 35 to 45 degrees centigrade. This can be seen mostly in the sugarcane, maize, etc. Okay, so in this, what happens? And here we should not write all the differences what we learned in the previous question, that is C3, C4 differences. Whatever are there in the C4 differences, C3, C4 differences, that part if you have written, you will get only half mark. Because here we are saying about the cycle. What are the steps that are present in the cycle you need to mention, not the plant characteristic features, what we are discussed in the previous answer. Okay, understood? So here the C4 cycle occurs in the mesophyll tissue of the leaf. That's the third point what we have read. So the first and the second points are the C4 cycle is also called as the hatch and slack cycle, which is going to be in the rice and the sugarcane plants and the maize. Okay, best examples are the maize, sugarcane, etc. And these plants can tolerate the temperature up to 35 to 45 degrees centigrade. Now comes the complete path cycle characteristic features, what we have to say. This C4 cycle occurs in the mesophyll cells of the leaf, where it is the mesophyll cells are having chloroplast dimorphism in them. It is having chloroplast dimorphism. So what is meant by chloroplast dimorphism? Two types of chloroplasts are seen in the leaf. There is a chloroplast dimorphism, and this occurs in the mesophyll chloroplast and bundle sheath chloroplast. In two chloroplasts, we will see this C4 cycle is going to be occurring. So it starts first in the mesophyll chloroplast and goes into the bundle sheath chloroplast and again comes back to the mesophyll chloroplast. In this, we can explain this like this. This is the Mesophyll. Wait, you cannot see if I write small. This is the chloroplast. This is also chloroplast. Okay, this is mesophyll cells. This is bundle sheath. So what happens in this? Which one will start this? The first one which starts this cycle is the PEP. So PEP is the phosphoenol pyruvic acid. This first accepts the carbon dioxide in the form of HCO3 minus and converts itself into the oxaloacetic acid There is a 4-carbon compound. So the first stable compound that is formed is the oxaloacetic acid. Based on this, the first stable compound, the cycle was named as the C4 cycle. And now in this, the enzyme utilized is the PEP carboxylase enzyme. Now this oxaloacetic acid converted into malic acid. And here it is the malic dehydrogenase is the enzyme. Okay, so here what happens? NADP H2s are reduced into the NADP plus H plus. Okay, and this malic acid, whatever is produced in the mesophyll chloroplast, will be going into the shifted into the bundle sheath chloroplast. Now, in the bundle sheath chloroplast, this malic acid will be converted into pyruvic acid. Okay, and here the carbon dioxide is released, that is the decarboxylation occurs over here. Once the decarboxylation occurs, this bundle sheath cells chloroplasts are having large amount of the rubisco enzyme in them. They are having the large amount of rubisco enzyme. Whenever the rubisco enzyme is present more, then this carbon dioxide was taken up by the RUBP, ribulose 1,5-bisphosphate, and goes into the starts the C3 cycle and converts it into the first stable compound as the phosphoglyceric acid, and again the cycle will be continued over there. Like that, the C3 cycle continues in the bundle sheath chloroplast, and at the same time, one molecule of water is released. Okay, now here the pyruvic acid, whatever is formed in the bundle sheath chloroplast, was shifted into the mesophyll chloroplast, and this pyruvic acid, when the ATPs are converted into ADPs and the inorganic phosphate that was taken up by the pyruvic acid to form the phosphoenol pyruvic acid in the presence of the enzyme enolase. It is giving the enol compound over there. So like that, the pyruvic PEP is going to be formed from the pyruvic acid. So like this, again the PEP is going to be regenerated in the cycle. So it's in the completely cyclic form. So C3, C4 is a cycle. Okay, like this we need to explain this, and after that we have to go with the other part of the explanation. The bundle sheath chloroplast are somewhat bigger than the mesophyll chloroplast, and these are arranged like a garland-like arrangement in the bundle sheath cells. The bundle sheath cells are the colorless cells which are somewhat bigger in size. And these chloroplasts are present like the garland-like occurrence. So this is called as the trans anatomy. And this can be seen in the few plants which are showing the somewhat xerophytic adaptations in them. Example, sugarcane and the maize. Okay, so this is the one what we have to write. This is the trans anatomy. So the C4 cycle we need to explain, and the Kranz anatomy we need to explain. We need to draw. Then we have to wind up this. And here inside this bundle sheath cells, we have to go with the xylem and the phloem. Because bundle sheath, that means the vascular bundle, should not be kept empty at any cost. This is the xylem. And this is phloem. These are bundle sheath cells. Like that, we have to draw the diagram and we have to show the Kranz anatomy and the vascular. But inside the Kranz anatomy, always, whenever we are drawing the stellar region, it should not be empty. So fill up with the phloem and the xylem. That's it. No further things. We need to go with this. That is the describe about the C4 cycle. Very easy. Only 4 steps will be there in the C4 cycle. But the explanation only need to be learned in a proper way. In the C4 cycle, C3 cycle occurs in the bundle sheath cells. Okay, where the bigger chloroplasts are there, where the RUBP is present, but Rubisco enzyme is abundant over there, so that the C3 cycle can start in the bundle sheath cells of these plants. That's the one we need to say and wind up this. That is the C4 cycle. Now comes with the next one. That is the most important question. Describe in brief photorespiration. What is meant by photorespiration? First of all, and how to write this, and in which chapter we will get it. Respiration is a separate chapter. When we don't know this answer also, at least we can score 2 marks or 2.5 marks without any stress on the answer what we are now going. If you forget this answer also, you can get the 2.5 if you write the relevant points. Okay, that is the photorespiration. That means inside when the presence of the light, respiration occurs. Photo means light now. So first to split the definition. Make the definition by splitting the word itself. Photo means light, respiration means exchange of gases. Okay, so in the presence of light, when the respiration occurs in an organism, that is called as the photorespiration. So this photorespiration is commonly seen in the C3 plants. Commonly seen in C3 plants. These C3 plants will be having the only C3 cycle in them. That means more amount of the carbon dioxide is needed for them. Okay. So how these plants will do the respiration part in the presence of sunlight? Whenever much more oxygen is there in the surrounding atmosphere, then the C3 plants will switch over into the photorespiration instead of photosynthesis. Why? Because whenever they need to do the photosynthesis, they need to have carbon dioxide in a purest form. Pure carbon dioxide in a much more concentration. Without that, they cannot start the photosynthesis itself. So whenever oxygen quantity is more, then carbon dioxide will be less in the outside atmosphere. Now, at that time, they will start the photorespiration instead of photosynthesis. Okay. So in this, the same enzyme is going to be utilized for the photorespiration. This photorespiration is also called as the C2 cycle. Why? Because the first stable compound is a 2-carbon compound. So we will call this C2 photorespiration as a C2 cycle. Okay. So this photorespiration occurs with the same enzyme, that is the Rubisco enzyme. So what happens in this? The first one is when the gaseous exchange, through the gaseous exchange, when oxygen enters in the stomatal apparatus of the cells and the chloroplast, when the oxygen concentration is more, then the Rubisco enzyme and a small amount of carbon dioxide is present. But the Rubisco enzyme is having the this enzyme is having 2 enzymatic grooves. Or what we can say, activation sites. Two activation sites. So in this, one is having the 2 different shapes also, these 2 activation sites. One is for the carbon dioxide and another one is for the oxygen. Whenever the oxygen is more and the carbon dioxide is less, then the oxygen acts as a competitive inhibitor over here. So what we learned in the previous class, enzymatic inhibitors, competitive, non-competitive, and the feedback inhibitors. So here oxygen acts as a competitive inhibitor and goes and blocks the enzymatic groove, otherwise the activation site of the Rubisco. So that the carbon dioxide which is present in a less quantity also, it cannot come and block this activation site of this enzyme. So the product formation not in a proper way according to the photosynthesis. Now once the oxygen molecules blocks the activation site, then immediately this Rubisco enzyme will start converting it into the phosphoglycerate and the phosphoglycolate. Okay. So it converts the ribulose 1,5-bisphosphate into phosphoglycolate and phosphoglycerate. This is 3-carbon compound and this is a 2-carbon compound. So in this, 2 products are going to be formed. One is the phosphoglycolate, another one is the phosphoglycerate. And in this phosphoglycerate, that is the phosphoglyceric acid, PGA, this won't start the further steps. The further steps were started with the phosphoglycolate. That is a 2-carbon compound. So we will call this a photorespiration as the C2 cycle. Okay. Now this phosphoglycolate will go into the different forms, into the different organelles. That is, it goes into the peroxisomes, mitochondria, and comes back to the chloroplast of the leaf, so that it will be going into the different forms and comes back to the reoccurrence, otherwise the regeneration of the ribulose 1,5-bis phosphate from the peroxisomes. Okay, so like that, this complete process is going to be occurred. And in this, what happens during the daytime when the photosynthesis not occurs in the plant body, means the starch preparation, that is the glucose preparation, is not going to be seen. With this, the energy is not going to be produced. And this is the completely, now it is going with the respiration process. The respiration always requires energy. The respiration always requires energy, so that the ATPs are going to be consumed more for this photorespiration process. So, which is a wasteful process during the daytime for the plant body. Only during the daytime, not for every time. So, during the daytime, the most important function, what it has to do is the formation of the glucose molecules. But here, instead of that, it is making the phosphoglycolate, which goes into the peroxisomes and the mitochondria for the respiration process. So ATP consumption will be more over there. So with this, completely the energy is going to be consumed in the daytime instead of producing the energy. So it is considered as a wasteful process for the plant body during the daytime. That is the most important one in this. And during this process, there is no synthesis of ATPs nor NADPH2s. No ATP synthesis nor NADPH2. There is no formation of this. With this, no energy is going to be gained for the plant body. So this much you have to write in the photorespiration. Then you will get 4 out of 4 for this question. Okay, now we will go with the next one is the hormones, plant growth hormones. I think I have not taken the VSQs also. Next one is the plant growth hormones. So, in this plant growth hormones, what the questions we need to learn? Here, how the auxins are useful in the horticulture and in the agriculture. That's the question what we have to say in this. Okay, and the uses of the gibberellins in the horticulture and in the agriculture. And next comes the cytokinins, ethylene, and the seed dormancy. In the March exam, you have seen they have given the seed dormancy, isn't it? So now we can expect the, that is from that type of questions, one question, and otherwise only based on the hormonal actions. So the first one is the for the auxins. Auxins which are useful in the agriculture and the horticulture. So when we are seeing this, how to write this answer is the most important one. So how to start this answer and how to end up beautifully this answer. So the auxins are the growth promoting hormones. Okay. So what we have to start, the first point in this is the auxins are the growth promoting hormones. Growth promoting hormones which acts based on the sunlight. So these are called as the PGRs, plant growth promoting hormones. Okay, phytohormones, what we can call them as the phytohormones, because they will act according to the outside sunlight. Now comes with the agricultural usages and the horticultural usages. So here what we have to write, the first one in the auxins, we know that this is a growth promoting hormones, and the usages will be the first one, it increases the cell enlargement. Okay, it helps out in the cell enlargements. So otherwise, the very first one, we can say the apical dominance. Apical dominance. What is meant by apical dominance? It suppresses the other hormones which promotes the lateral growth in the plant body and makes the apical bud to be always dominant. That is called as the apical dominance. That means linear growth of the plant body can be seen, rather promoting the branch formation. That is the apical dominance. Okay, and the next one is with this, the stem length can be increased a lot, and the branches, sub-branches, and the other parts are going to be less in the plant body. The formation will be less. And the next point. Just a second. Sorry. So in this, the apical dominance means only the length of the stem is going to be increased by decreasing the branch formation in the plant body. That is the apical dominance. Example, we can see in the sugarcane plants, where the branches are very less or none, we can say, and only the stem length will be increased. And the next one is it enhances the cell division. Enhances the cell division. Here you should not say increases, it's the enhances. If you write the word as enhances, then your standards will be maintained in answering this type of questions. Okay, where the theoretical questions and the prominent words need to be used for this part, where the word is the enhances. That means drastic increase, enormous increase. That is the enhances the cell division. That means it makes the cells to divide, redivide, divide and redivide many times faster than any other hormone. With this, new cells are going to be formed, and the next one, growth will be faster enough, as well as the resistance capacity will be increased in the plant body. So like that, with this two ways, the plant is going to be benefited. Resistance will be increased, as well as the length of the plant body is going to be increased. With this, the leaf promotion can be done more. Okay, and the resistance capacity, so that the product, that is the yield, will be more from this plant body. That's the most important one, what we have to conclude, because they are asking with the agriculture and the horticulture. So when we are saying this examples, along with the examples and the explanations, then that will be the best method. Okay, so with this, with the enhancement of the cell division, leaf promotion will be more, as well as the disease resistance capacity will be increased in the plant body, so that the yield will be 4 times more from the plant body. That is, in the agriculture also we can use, as well as in the horticulture. In the agriculture, if we use it with the sugarcane, then the stem length will be more and the leaf will be more. With this, the sugar formation, the thickness of the stem will be more, sugar contents will be more for us, more juice, more sugar. And after that, we will be having the in the horticulture. In the horticulture means the fruit yielding plants. The yield will be 4 times more than the other plants. So we can get the more cash. Okay, encashment will be done in a large number. That is the second point. And now comes the third point. It elongates the internodal length. Internodal length will be increased. The third point with the auxin says internodal length is going to be increased. Internodal length is increased. Okay, like this, when the auxins are used, the internodal length will be more. Whenever the internodal length will be more, the stem elongates. Okay, in between the two nodal regions, that is the internode, which is having the more distance, that is the more length. So that is the internodal length is going to be increased. Example, sugarcane. Okay, and the next one is it enhances the parthenocarpy. So what is meant by parthenocarpy? We have already discussed this. Without the fertilization, when fruit formation is there in the plant body, that is called as the parthenocarpy. So by the increase of the parthenocarpy, what happens in the agriculture and the horticulture? In the agriculture, we won't use the parthenocarpy, but in the horticulture, we'll use the parthenocarpy. That is for the production of the grapes, citrus fruits, apples, where we can get the much more cash of selling of these fruits. So what we have to write, by the increase of the parthenocarpy in the horticultural area, where the fruit promotion will be more with the more juice in it. That is nothing but the example, grapes and the apples. Okay, all the seedless fruits are going to be made with the parthenocarpy. And the next point is by the application of the auxins, we can change the sex of the flower. That is, the male flowers can be converted into female flowers. So with this, the more production of the fruits can be seen. That is the example cucumber. So this is helpful in the hybridization process also, and as well as with this we'll get the much more amount of the fruits. That is the sex determination. Okay, this is also with the auxins. And the next one will be the auxins. And auxins can be used in the biotech labs for the in the tissue culture. So along with cytokinins, auxins will increases the root and the shoot promotion of the tissues in the tissue culture lab. So that is the one what we have to say. In the horticulture, we'll go with the tissue culture for the development of the new plants, so that it can be easily grown from the meristematic tissues only. No need of seed to be developed into a new plant. Okay. So in that case, auxins should be more and the cytokinins are less. Then it will be promoting the root formation. Auxins and cytokinins. Root formation when auxins are more and cytokinins are less. Okay, when cytokinins are more, auxins are less, then it promotes the shoot formation in the tissue culture labs. So like this, we can write the, and it enhances the flowering. It enhances the flowering in the some plants. Example, grapes. So that the parthenocarpy going to be increased in the grapes. So like that, we can conclude the auxins in the which are useful in the agriculture and in the horticulture. And the next one is the gibberellins. Gibberellins. Gibberellins are the growth promoting hormones, which are also considered as the phytohormones. These gibberellins are going to be synthesized by the matured buds and stored in the another part of the stem, so that the internodal length will be increased. Okay, so the gibberellins are the phytohormones which are going to be synthesized by the matured leaves and the fruits and stored at the nodal regions of the stem. And these gibberellins, the next point will be the gibberellins removes the genetic dwarfism. It removes the genetic dwarfism. Removes the genetic dwarfism. Example, in the cabbage, in the Brassicaceae family members, which will be by applying the gibberellins, the internodal length will be increased. So the dwarf plants become the normal sized plants. That is the removal of the genetic dwarfism. And the next one is it increases the internodal length. Increases the internodal length. So that the plant gets the good height. Okay. And the third one, it enhances the parthenocarpy. It enhances the parthenocarpy in tomatoes and in Solanaceae family members. It enhances the parthenocarpy. That is, seedless fruits are going to be formed by the application of the gibberellins in some plants. Example, tomatoes and the grapes. Okay. And the fourth one is sex determination. It changes the sex of the flowers in some plants into the female flowers into the male flowers, so that it will be very much useful in growing the rose flowers. Okay, and in the pineapple also it can be used very easily for the production of the pineapples. And the next one is the remove bolting. Without this, we can go with some useful one, which you can easily remember. Okay. And the fifth point in the gibberellins is what I can say for you very easily. It removes the seed dormancy in monocots, especially in the monocot plants. Example, rice, wheat, etc. In them, if we treat the seeds with the gibberellins, then they remove the seed dormancy period and makes the seeds to be shown the growth very fast. Okay, that is the only with the gibberellins it can be possible, and it removes the boltism. That is the buds are going to be shown the proper growth by the application of the gibberellins in them. And the internodal length will be more. And these gibberellins are going to be synthesized in the older buds as well as in the older parts of the leaves and stored in the roots. So that is the phytohormones, what we can say. Whereas auxins are synthesized by the roots and stored at the tip of the stem. So that is the most, and this gibberellins will enhances the cell division. Enhances the cell division. In some plants, it increases the cell division at the nodal regions so that the leaf promotion and the bud promotion will be more, as well as the cells will be more at from the nodal region, so that the length of the stem will be increased a lot. These are the ones what we can say for the gibberellins. So practice these questions what we have discussed till now in these classes, so that we will be getting the much more importance of these questions, and we will get maximum from this part, and we will see the other questions that are the cytokinins, ethylene, and the abscisic acid, and the seed dormancy from this. And one more question, we cannot guess that from the respiration we'll get a question, but chances we cannot say. So we will prepare by the end of this, and after this we will start the VSQs from the 9th chapter, 10th chapter. Okay, most important VSQs also we will start doing from the next class onwards, so that we can wind up the complete syllabus. Okay, so today is the Monday. So next Monday onwards, you are having the supply exams. So prepare well from these concepts till now, what we have discussed. So that what we have to prepare for the supply exams, which chapter, the further LQs. I am saying again, so that you have to prepare thoroughly with these chapters. Chapter 13 and 14, that is one LQ we will get. sewage treatment, or else the plant breeding process, or else the tissue culture. Okay, out of these three, one will be mandatory for the LAQs. Next comes the SAQ chapters. Which chapters you have to concentrate for the LAQs? Chapter 7 and 8 for one SAQ. That means from these two chapters, we will get one SAQ and one VSAQ. So prepare well with the bacteria and the viral chapters. Okay. Next comes the chapter 9. That is genetics. Again, one SAQ, one VSAQ. Okay. Mandatory from these chapters, beta. No chance of not giving the question from the chapter 9, SAQ and the VSAQ. And from these two chapters, one SAQ, one VSAQ. And after that, we have to go with the chapter 12. For one more SAQ and one more VSAQ, that is applications of biotechnology. That is also most important one. Okay. After that, we have to, till now, we have finished with the 3 SAQs. Now start going with the chapter 1, 2, and 3. That means we can wind up with the 2 SAQs or at least 1 SAQ, and VSAQs, if you prepare, you may get a VSAQ also. So always it will be easy, and we damn sure we will get a question from these 3 chapters. So like that, how many SAQs we have finished? 1, 2, 3, 4 SAQs we have finished. 4 4s are 16. And 3 VSAQs. 16 plus 6, 22. Pass. Pass. And 1 LAQ. 8 marks if you take. 22 plus 8. Or else 7, 29. Okay. If not 28, take out 1 more mark with the spellings or any other thing. That is also very important. And the chapter 11 and chapter 14 and what else? Chapter 10. From this chapter, 2 VSAQs we will get. And chapter 11, 1 VSAQ we will get. So there 27 na 28 na 28 plus 6, 34, if not 32. Bindas 50% marks we got it, isn't it? So prepare well what we have finished till now. Okay, so that easily we can cope up with the supply exams and no need to worry. Actually, if you follow this pattern and the weightage, damn sure everyone will pass. Okay, but if whatever I am saying, you have to believe me and read the answers also. That is also mandatory. The next work, what we have to expect is you have to prepare. If not, nothing can be happened in the life. So the preparation is mandatory at home, and you have to clear. And when you have to clear this exam means you need to prepare well. And in this, don't say that important questions again, because chapters are the important ones. In each and every chapter, we are having nearly maximum 4. And only in the first chapter and in the 9th chapter and in the bacteria and the virus together now. So no need for us to worry. Okay. So like that, in few chapters only we are having nearly 8 questions. But in all other chapters, we are having 3 or 4. That much only. So we can prepare with that. In that 4, one will be coming means we need to prepare. And here we cannot expect that this question will come damn sure. From this chapter, one question can be expected, but not more than this. So prepare what I set the weightage for you right now. Okay. So don't be lazy. Don't roam on the roads and start preparing well, at least from now onwards for the completion, for getting the at least 50% marks. Okay. What if your efforts of the 2 years will not be wasted at any cost. Okay. So tomorrow we'll meet at the same time, same channel with the few more SAQs and more VSAQs where you will get damn sure questions from those VSAQs. Okay, right? Thank you.